Even more scary these days is the rise of multidrug-resistant bacteria, so we need creative solutions to fight them. Imperial College London and the University of Nottingham have formed a corporate research group that has made experiments using a predatory bacterial species, which could mean a whole new angle in protection against antibiotic-resistant pathogenic bacteria. As a first step, they have examined whether a predatory bacterium called Bdellovibrio bacteriovorus destroys Shigella flexneri. Shigella is a bacterial species that causes food poisoning and is responsible for 163 million cases of illness and over 1 million deaths annually. The test was successful, so they continued the process with animal tests. For those, they used zebrafish larvae. One group was infected with Shigella only. The other group was injected with Shigella and Bdellovibrio as well. The presence and activity of Bdellovibrio promoted zebrafish survival without exerting pathogenic effects on the zebrafish larvae.
Bdellovibrio are motile bacteria, so they swim around and they are able to locate a prey bacteria. And so they attach, so they're very small, so they attach onto the surface of the prey bacteria, then they invade it. And as they invade it and get inside, it rounds up the prey bacteria. So something that was rod-shaped becomes round. And then it uses enzymes to eat the prey bacteria from the inside, and so it uses all of the energy and the mass that it takes from the prey to grow, and then once it's fully grown it divides and lyses out of the cell so it can go infect more prey bacteria. So it invades a wide variety of gram-negative prey bacteria.
In my lab, we focus on Shigella flexneri, which is an important pathogen, which also happens to be a gram-negative bacterium. And so because we spent years really perfecting this Shigella zebrafish infection model, it was the first opportunity for us to test Bdellovibrio, its mode of action in a whole living animal model. It's been known for decades that Bdellovibrio can invade and replicate inside gram-negative bacteria in a lab, in a test tube. But what the zebrafish model uniquely offered us the opportunity to study was to go under a microscope and to watch how does Bdellovibrio work in the context of a whole animal model with a fully functional innate immune system. We showed we can put in as much Bdellovibrio as we want into the fish, and it was perfectly safe. The fish showed no ill effects, they didn't get sick, they didn't die, and what was important is that the zebrafish eventually cleared it. It offered a window of therapeutic opportunity. Bdellovibrio stayed alive long enough, in this case, to fight Shigella infection.
What was great about the zebrafish was that it's also got its own host innate immune system, and so we could test Bdellovibrio predation of Shigella in the context of an innate immune system. And so what we saw was host recognition of Bdellovibrio. So the host can clear Bdellovibrio slowly over time. So once they run out of food, they can't replicate anymore. They're absolutely dependent on prey bacteria to replicate. They actively get eliminated by host innate immune cells like macrophages and neutrophils.
The key message is that it could synergize with our innate immune system to clear multidrug-resistant Shigella. It still worked in the absence of an immune system, which is important for safety, but it was in the presence of an immune system that it really worked best. And that's a kind of a different way to think about fighting multidrug resistance. Some of the key questions that we're now in the process of addressing would be, does it work also well on other bacteria? So in this case, we showed Shigella. Does it also work on different types of gram-negative bacteria? I mean, that was the premise of the paper, is we need creative solutions to fight bacteria which are multidrug resistant.
Zebrafish larvae are a perfect model system for basic research. But in order for Bdellovibrio to ever be used in humans, additional experiments are needed in higher vertebrates, for example in mice. In this paper, the authors showed how Bdellovibrio worked in a compartmentalized infection in the hindbrain. But the next question is whether this also works in other bacterial infections. There are many questions to be answered before researchers can start clinical trials in humans. But maybe one day this little microbial knight can fight in your body against E. coli and Salmonella infections as well.